Hi, my name is Aing. I am a first-year student at Soka University of America. I am presenting to you a research on genetics. This was done as a part of an introductory biology class in my first year. Me and my classmates have done this over a course of 8 weeks with the help of our professor. Our goal is to discover the effects of certain mutations on chromoproteins color expression. Here's our abstract. So our research was based on a previous paper about chromoproteins. From this paper, we learned that chromoproteins come in a variety of colors, as you can see here. They are very easily detected in visible lights, which makes them efficient biological markers. We also learned that the chromophore region affects chromoproteins' color most strongly. In the paper, the author did random mutations at the chromophore of the protein amilCP, which resulted in different changes in its color. We wanted to do the same thing with a different chromoprotein, aeBlue. So in order to generate new colors from the aeBlue, we had to come up with a novel idea for our site-directed mutation. So after the journal club discussion, we had to design a primer. This could either target the amino acids within the chromophore or the ones surrounding it. We then had to explain our decisions and give prediction of the possible results, such as a loss or a shift in color. Finally, we needed to provide a screenshot for our proposed primers. With a view to observing a change in its color expression, we had two approaches: site-directed mutagenesis and random mutagenesis. For site-directed, we used the design primers that I mentioned in the last slide, and for random mutations, we use sloppy PCR. We then transformed those into bacteria. We isolated the DNA and sent them for sequencing. We also visualized the proteins using the SDS-PAGE. The transformed bacteria were put on agar plates. Here you can see that all the site-directed mutagenesis turned white, which means that all the mutations have affected chromoproteins color expression. Here you can see the chromophore CMYG starting at position 61. Some primers targeted the amino acids in the chromophore, while the others targeted the ones surrounding it, as you can see here. We aligned the sequences and saw that 5 out of the 7 yielded the mutations that we wanted. Our random mutagenesis, delta 159, had a nonsense mutation, which left it with 159 amino acids. This one also lost color. When we analyzed the proteins using SDS-PAGE, we observed that other than delta 159 and C62H, all the other missense mutations had the same size as that of the wild type. Delta 159 and C62H, however, were not detectable. So in conclusion, we were able to do 5 out of the 7 plant site-directed mutants. All of those lost color. There's also one random mutant with a nonsense mutation. Our mutations, however, were not sufficient to change the color of aeBlue beyond a loss of function. In the future, we hope to make more site-directed mutagenesis and maybe combine the mutants that we already had. We could also do more experiments to confirm that whether the most abundant band is in fact the protein of interest. We could also try using different chromoproteins other than aeBlue, which might be more stable in color expression. Lastly, we can layer this exercise on top of protein modeling and folding software. We want to express special thanks to the rest of our classmates from Bio 140 class, and thank you for listening.